Anytime we perform an experimental manipulation, we have to be careful that the techniques that we use to perform our manipulation don't have some unintended consequences. So, for example, when Leslie Lancaster et al. Manipulated estradiol concentrations in some lizard eggs by adding a little bit of estradiol to the outside of the eggs, she needed to be careful to control for the fact that that estradiol was dissolved in ethanol as a vehicle to deliver the estradiol. And as a result, extra care needs to be taken to make sure that it wasn't, in fact, the ethanol rather than the estradiol that caused the changes in back patterning. This is true anytime we perform an experimental manipulation. We will always want to manipulate only the factor of interest, but oftentimes in order to manipulate that factor, we need to do something else at the same time. So, for example, I was part of a study that miniaturized hatchling lizards by experimentally removing some of the yolk from the egg with a syringe. But in addition to removing the yolk, this procedure also involves piercing the outside of the egg with the needle. And so, as a result, if we want to test what the effects are of removing yolk from the eggs, we need to not only have eggs that are completely untouched, eggs from which we have removed some yolk, but we also need to control for the poking itself by taking some eggs, poking them with the syringe, and then removing the syringe as a control for the fact that we have pierced the outside of the egg in our yolk removal experiment. This is what's referred to as a sham manipulation, where we perform all of the same techniques as our experimental manipulation without the factor of interest. Sham manipulations are appropriate whenever our experimental manipulation has some other unintended consequence. So, for example, we might be interested in warming temperatures in tundra ecosystems using open-top chambers, but those chambers might also affect snow accumulation that we might need to control for through a sham. Sham manipulations are particularly important when humans are the subjects. That's because of what's referred to as the placebo effect, or the real or perceived effect of some treatment, of just being told that you're participating in an experiment that has some benefit. So, for example, 30% of human subjects report receiving some pain relief after being provided with pills that they expected to relieve their pain. So in cases where we're trying to test the efficacy of some drug, we not only need controls and experimental subjects who receive the treatment of the drug, but we also need to provide some individuals with a placebo, a sugar pill that doesn't contain any of the active ingredient, but would otherwise convince the human subject that they are receiving the treatment. So we need to acknowledge the fact that humans are not completely objective subjects. And this can occur not only in experimental scenarios, but also in observational scenarios. The Alberta Biodiversity Monitoring Institute measures biodiversity at 20-kilometer intervals throughout the province of Alberta to try and determine how human land use is affecting biodiversity in the province. Now, these are at regular 20-kilometer intervals, but the exact locations of the sampling sites are confidential, because if they were known, humans might change their land use as a way of manipulating the changes in diversity that have been documented, or potentially to influence the future outcomes of this monitoring project. In addition to not being objective subjects, humans are also not completely objective observers. Sometimes our data collection can be influenced by our knowledge of what treatments the organisms have experienced that we are trying to collect data from. As a result, this can introduce bias into our measurements, either because of our preconceived expectations about how organisms should respond to treatments, or in some cases, a reverse bias, where we're so concerned about biasing the data in the direction that we think the results will go, that we actually cause a bias in the opposite direction that might counteract any effect that we're trying to document, just because we want to be extra careful to not be biased. The solution to this problem is to make sure that observers collecting data are always unaware of the treatments that those subjects have experienced prior to the measurement. So in summary then, we identified that anecdotes are not particularly strong forms of scientific evidence. We defined experiments to be situations where the researcher has manipulated the conditions experienced by some subjects and not others. We identified some potential pitfalls associated with experiments where the researcher might need to take particular care to not confound the experimental manipulation with some aspect of the technique or vehicle used to implement that manipulation. And finally, we talked about the various ways in which humans might be biased, either as subjects or observers, that we ought to be particularly careful of in designing our experiments.